Welcome to the third part of our lecture on the characteristic features of the phylum Arthropoda. For the outline of the presentation, the first part of the lecture will focus on the nervous system of the arthropods. We also have the digestive system of the arthropods, their excretory system, and their reproduction. For the overview of the classification of arthropods, so arthropods are invertebrates, and we have two classes of veterinary importance under the phylum Arthropoda, and those are the class Arachnida and the class Insecta. For the nervous system of the arthropods, so the arthropods have a brain and a ventral nerve cord containing clusters of neurons called ganglia in segments. So the diagram shows the nervous system of an insect, which is a class under the phylum Arthropoda. So in this diagram, it shows the location of the brain, as well as the ventral nerve cord and the segmental ganglia. A peripheral nervous system, or PNS, connects the CNS with the rest of the body. The central nervous system of the arthropods, particularly insects, is simple and consists of a series of ganglia that supplies nerves to different segments of the body. It consists of a dorsal brain, which is adjoined to a ventral nerve cord. The ventral nerve cord has paired ganglia, which run along the midline of the thorax and the abdomen. Since the body of the insects is segmented, each segment has ganglia, which are linked to a commissure nerve and joined by intersegmental connectives. The commissure nerve is a short medial nerve. Thus, the central nervous system of insects has a ladder-like appearance. So we have here a diagram showing the representation of the insect nervous system, showing a dorsal brain. We also have the series of ganglia. For the thoracic portion of the body of the insect, it is known as the thoracic ganglia. And for the abdominal portion, we also have here the abdominal ganglia. The ganglia act as a control center for the body sections in which they are located. We also have here another diagram of the different parts of the body of the insect, showing the nervous system of the insect in blue. So we have here the brain, the ventral nerve cord, and the segmental ganglia. It is important to study the nervous system of insects because the nervous system is considered to be the target for many insecticides. The sense organs like auditory organ, we also have the statocyst, tactile organs, eyes, sensoricity, and the antennae are also present in the arthropod. The next is the digestive system or the alimentary canal. The alimentary canal of the arthropods is composed of three parts. First part is the anterior stomodeum or the foregut. We also have the mesenteron or the midgut and the posterior proctodeum or the hindgut. The alimentary canal of insect is considered to be long, muscular, and tubular structure extending from the mouth to the anus. It is separated into three areas: the foregut, the midgut, and the hindgut. Again, the anterior part of the alimentary canal may have pharynx. So it contains a pharynx. At times behind this is a dilatation called the crop or the proventriculus is present, which in some species is followed by the gizzard. The proventriculus act as a valve to prevent regurgitation and may have teeth to aid in the disintegration of the food particles. So this is exemplified by fleas. This leads to the mesenteron or the midgut. It is here that the food is digested and absorbed. The proctodeum consists of the anterior ileum. We also have the colon, the dilated rectum, and the anus. It opens out into the exterior through the anus. The main function of the hindgut or the proctodeum is the absorption of water from the feces and urine. The urine passes into the hindgut from the malphigian tubules, as shown in this figure, which open just behind the midgut. The feces are excreted as dry pellets and nitrogenous waste as uric acid. We also have here a longitudinal section of the alimentary canal of the insect or the arthropod, showing the three segments or the three parts of the alimentary canal: the foregut, the midgut, and the hindgut, with its associated parts. We also have here an anatomical view of the alimentary canal of Gamsoclis gratiosa, which belongs to the order Orthoptera. So in this anatomical view of the alimentary canal, we can see here the segment or the section of the foregut now containing the this portion here. We also have the midgut and the hindgut. For the excretory system, the insect excretory system consists of malphigian tubules and hindgut. The malphigian tubules, which are arranged in a ring around the alimentary canal at the junction of the midgut and the proctodeum. So the insect excretory system plays essential roles in osmoregulation, ionoregulation, and toxin elimination. Understanding the mechanisms of fluid and ion transport by the epithelial cells of the excretory system provides a foundation for the development of novel pest management strategies. This diagram shows the location of the malphigian tubules of the insect, which is considered to be the main excretory organ of insects. We also have here the coxal glands. So the coxal glands is one of the certain paired glands with duct openings in the coxal region of the arthropods, particularly the arachnids, functioning as excretory organ. So this diagram shows the location of the coxal glands of the arthropods, particularly arachnids. So for arachnids, one of the main excretory organ is the coxal glands. So the word coxal comes from the Latin word coxa, meaning hip. And these are common among, the coxal glands are common among arachnids. When we say arachnids, this refers to the spiders, scorpions, ticks, and mites. The waste are collected from the surrounding hemolymph of the hemocoel and discharged through pores on one to several pairs of appendages near the proximal segment of the leg. Other arachnid species have malphigian tubules instead of or in addition to the coxal glands. In some of these species, however, malphigian tubules seem to function in silk production rather than excretion. Spiders may use malphigian tubules or coxal glands for excretion. So this diagram shows the external organization of a spider, showing the location of the coxal glands. We also have here the internal organization of the spider, again showing here the coxal gland, as well as the malpighian tubules, which are the excretory organs of the spiders. Another structure is the fat body. So the fat body is a large structure made up of cells containing numerous fat vacuoles. Its form is variable, but in many insects, it lines the body cavity and all the internal organs of the insect. So it is analogous to the peritoneum in mammals. The fat body has many metabolic functions and act as a food reserve for use during periods of hibernation or starvation. So we have here the location of the fat body of the arthropods or the insects. Aside from that, it also absorbs fat-soluble insecticides, reducing their potency. For the reproduction of the arthropods, so most insect species have separate sexes. The reproductive tracts are directly analogous to those of the vertebrates. That is, they also have the testes, the vas deferens, and the seminal vesicles for the male, and the ovaries, the oviducts, the common oviduct or the uterus, and the vagina for the female. This diagram shows the generic scheme of the female reproductive system of the insects, showing the location of the ovaries. We also have the oviduct, the common oviduct or the uterus, and the vagina. We also have here another representation of the generic scheme of a male, the reproductive tract of a male insect. So the reproductive tracts are is considered to be analogous to that of the vertebrates. So in the male insect, they also have the presence of the testes. They also have the vas deferens and the seminal vesicle, as well as the penis. For male insect, we have the term edegus. The edegus is considered to be the penis of the insects. It is the reproductive organ of the male insects. It is attached to the abdomen and used to deliver sperm into the female insect during reproduction. So this diagram here represents the edegus of the male Formea rehina. We also have here another diagram showing the reproductive tract of a male blowfly, showing the testes, the vas deferens, we also have here the ejaculatory duct, and the penis or the edegus. This portion here is the vas efferens, and we have here the vas deferens. Females also have an accessory organ, the spermatheca. The spermatheca acts as a receptacle for spermatozoa after mating. This enables the female to fertilize subsequent batches of eggs in the absence of a male. The figure shows the generalized structure of the female reproductive tract, showing the location of the spermatheca or the spermathecal gland. We also have here another diagram of the reproductive system of the female apple maggot, Rhagoletis pomonella, now showing the ovary. You also have here the common oviduct or the uterus, the spermatheca, here represented by the S. We also have the accessory gland and the bursa copulatrix and the vagina. For the grasshopper reproductive system of the adult female, We have here the presence of the ovariole or the ovaries, the oviduct, the spermatheca, the vagina, and the ovipositor. Most adult female insects are considered to be oviparous. So when we say oviparous, this is an insect that reproduces by laying eggs, and most of the eggs development occurs outside the mother. This is also known as oviparity or egg laying. In most cases, the female insect will lay eggs either singularly or in batches in a suitable place. For example, the females of some species of mosquito lay eggs in the form of a floating raft on the water. These rafts can contain more than 250 eggs. The diagram shows a female Culex mosquito which lays an egg raft on the surface of the water. This mosquito exhibits oviparity, or it is an oviparous insect because it lays eggs that will later develop in the external environment. Other examples of oviparous insects include the housefly or the Musca domestica and the blowfly of the family Calliphoridae. Some insect species are considered to be viviparous, that is, the egg ruptures at some stage within the reproductive tract, so the female gives birth to a juvenile. This mechanism is also known as viviparity. Viviparity means to give birth to live young rather than laying eggs. Most insects produce eggs, but some, such as aphids, are viviparous and give birth to live young. The female aphids reproduce by means of parthenogenesis. In species where viviparity gives rise to larvae, the species can be termed as larviparous. So the other name for viviparity include larviparous. This diagram shows the aphids. The aphids are considered to be viviparous and give birth to live young. There are several forms of viviparity in insects, but adenotropic viviparity only occurs in certain true flies of the order Diptera, such as the tsetse flies or of the genus Glossina. The insects that exhibit adenotropic viviparity have eggs that hatch within the female insect. The larvae are then nourished inside the female by special milk glands. The larvae remain in the female until mature. At this point, they are laid and immediately pupate. This diagram shows a pregnant female tsetse fly, Glossina morsitans, from a colony kept at the Yale School of Public Health in New Haven, Connecticut. This diagram here shows a comparative morphology of the dipteran reproductive organs. So when we say diptera, this refers to the order in which these particular insects belong. This tsetse fly and this Drosophila fly. So the letter A here represents the reproductive tract morphology of the Drosophila fly. We also have here in letter B, the reproductive tract morphology of the Glossina or the tsetse fly. The letter C here represents the magnified view of the Glossina milk gland tubules, which highlights the change in the tubule and secretory cell physiology in the transition between the distal and the proximal milk gland. The letter D here represents the magnified representation of the intrauterine wall and the choriotet structure. So when we are going to compare the reproductive tract of the tsetse fly from that of the Drosophila, which is a typical dipteran fly, we can see here that the tsetse fly has this particular milk gland in it, and it also has a different uterine structure compared to that of the typical dipteran fly, which is the Drosophila fly. This diagram shows the schematic representation of the first gonotrophic cycle of a Glossina morsitans female under optimal environmental conditions. When we say gonotrophic, that is the period between the blood meal and the oviposition. So oviposition means the expulsion of the egg from the oviduct. The different stages of the oogenesis, embryogenesis, and the larvae genesis occurs within the Glossina reproductive tract, particularly in the ovaries and the uterus. So this is a typical example of adenotrophic viviparity. We also have here the diagram showing the unique biology of the tsetse fly of the order Diptera. So it exhibits this particular characteristic, which is the intrauterine larval development. The females produce one offspring per reproductive cycle. The larvae are held in the female uterus for their entire development. The females nourish the larvae with nutritional secretions from a modified accessory gland called the milk gland. So the accessory gland for the tsetse fly is also known as the milk gland, and that is unique among these species of the dipteran fly.